Do you have the CD? Going through mastering, yeah. Huh. I should mention that to the publisher. Maybe they can do something. Okay, so as a fellow classmate just informed you, and for those of you who don't know what interactive physiology is yet, please take time to slap yourself. Okay, and then go use it, because it's an amazing study tool. And for this chapter, it's an amazing help, because it's gonna do a lot of the things that I do in class. I sort of compare information to other things to help you remember. And this does a really nice job of doing that for an immune response. So, phagocytes, specific or non-specific? Non-specific. They just know something sticky, it's stuck to them, they're going to eat it. Yes? Know the steps of phagocytosis. This is repeat. You know this information already, right? Say yes, Maria. We know this information already because we learned it when?
Last time.
We learned it last semester.
That's what I meant by last time.
Yeah. Okay. So when we talked about, I think it was in chapter 5, and we talked about inflammation, inflammatory response, we talked about phagocytes there. So phagocytes, non-specific. What are natural killer cells? We kind of left off talking about these guys in my Bad Bee movie last class. They're what? They're lymphocytes, and what do? Yeah, they use different methods to basically poke holes in things, poke holes in invaders to help protect you from foreign substances. We're going to talk a little bit about the substances some of them use, but they are typically, when you did your white blood cell differential count, and you hopefully took a look at some of the diagrams to help you figure out who was who, you notice that lymphocytes aren't always little dinky, almost all nucleus cells. That some of them are sometimes a little bit larger. That's these guys, the larger lymphocytes that might have a few little granules in them. That tends to be these guys, the natural killer cells. So they're not all going to be those small little lymphocytes floating around. These guys are floating around too to help protect us in a non-specific way from foreign invaders. We talked a little bit, and they talk a little bit, about inflammation and inflammatory response. Again, repeat information from chapter 5. So understand what triggers an inflammatory response. When I cut into tissue, and that's what happened to this poor little person up in the corner, their beaker in lab class was broken. They have a boo-boo. So tissue injury. Think of your cells as eggs. Crack it open, spill out its innards, and that's a chemical signal for an immune response. Again, this is non-specific, but those chemicals that are released from injured tissue, damaged tissue, are going to trigger a series of events that are going to cause cells to come to the area and help heal. So this is one of those psychotic diagrams that we see in the textbook. We've seen many of them throughout the semesters, haven't we? But usually they're really good information because they tie everything together for us. So when we injure tissue, that's the initial stimulus to this response. We're going to have some physiological response occurring, and that's what's in the green in this diagram. So we release chemicals from the cells, things like histamines, complements, kinins, prostaglandins. These are all chemicals inside the cells, and they're released when the tissue is damaged. That is going to induce something called leukocytosis, which is what? Yeah, it's going to increase the number of white blood cell and also do what? Attract cells to the area. And why do I want cells in that area? These cells in that area.
Start killing it again.
Yeah, specifically those phagocytic guys for that non-specific response to get rid of anything that might breach the skin, which I have cut into. The other thing that's going to allow cells to get to the area is increased capillary permeability. What the heck does that mean? And that's what causes inflammation, yep. Because what's going to happen when I increase capillary permeability, I can leak more fluid out of them. Fluid's going to build up in that area. But why does this happen? And these guys have to get out of where? They have to get out of the circulatory system and into the surrounding tissues. So this is kind of a result of that. This is what causes that pain and swelling. This is what can cause the heat and redness, just a buildup of tissue fluids in general. But that's important to get the cells to the area. All of this stuff is going to help do what? Get rid of debris. So migration of cells to the area, something called margination, where cells are going to cling to the side so they can leave. Then diapedesis, that's the actual me leaving the circulatory system. And then doing the job that I was meant to do, getting rid of any dead debris or nasty little critters that might get in the area. A buildup of these guys can form something called pus. Is all pus bad? No, but you shouldn't let it build up too much because it actually can act as a cover. So what should you do with pus? Try to get rid of it, rinse it away. Pus is all these guys dying, trying to keep you protected and ending up dying, which is okay. They did their job, bless their hearts. But don't let them build up too much. So you want to rinse them away so that new cells can come to the area and finish cleaning up debris. And that's what's going to help with the healing process. So they talk about that under inflammation and tissue response to injury. And that's the best diagram on page 768, figure 21.3, to look at for all of the different players in those steps. So we talk about getting phagocytes to the area under phagocyte mobilization. Leukocytosis, we kind of stick to the side. Margination and diapedesis. And we can see these steps listed for us on page 769, figure 21.4. Once I get into the tissues, what's going to tell me where I need to go next? What's the last step of this? Look at number 4. Read the big word, chemotaxis. And what's chemotaxis? Yeah, it's like a chemical trail. Remember, when those cells become injured, they're going to release their innards. And that's the chemical trail for these cells to follow. So neutrophils are going to be attracted to that damaged tissue because of the fact they are attracted to the chemical trail left behind by those dead cells. Typically in the area, especially when we have an injury like this, because we've breached that outer barrier, we're letting in foreign invaders. So it's smart that we're attracted to the dead cells because that's probably where, if they're going to get in, they will be. And they, I'm referring to foreign invaders that the neutrophils can then phagocytize and get rid of. So know the steps of phagocyte mobilization. Oh, what a good essay question. They are on page 769 in your textbook. Exactly. That's how you should think of an immune response, as an army. There's different players in the army. They have different jobs. They have different functions. And that's exactly what an immune response is. So, on page 770, there's 2 tables that give us a nice overview of some of the different inflammatory chemicals and some of the body's innate body defenses. What's innate? Non-specific. So it goes through some of the different innate defenses, those first-line defenses. We have our skin, our acid mantle, keratin. What's that? It's our waterproofer. Remember we learned in chapter 5 that keratin was a chemical that was given off by the cells, the keratinocytes, in your epidermis of your integumentary system. Remember that? Well, that also is a chemical that's going to help protect you from foreign substances as well. Mucous membranes and mucus in general help things stick to it. So these guys, these cells that we keep talking about under innate defense, have time to get to the area and dispose of the invader. So mucus, hairs in your nose, nasal hairs, cilia on your cells. We already had the disgusting smoking talk last year, right? We will have it again for those of you who missed it because it's very exciting when we discuss the respiratory system. So cilia on the pseudostratified ciliated columnar epithelial cells. I believe I said that all in one without a breath. And where do they line? The trachea are going to help protect you from foreign substances. Sweep up any of that stuff that gets stuck in the mucus that lines that tube as well. Gastric juices, when we talk about the digestive system, we'll see the pH is very, very low, extremely acidic, and it's going to help you as well. Acid mantle and mucous membranes like the vagina are going to help trap any foreign substances that might make it up that canal. Your tears, your saliva, urine. Do not be afraid of urine. Urine is sterile. Unless you are sick, urine is sterile. Unless you have a urinary tract infection, urine is sterile. Yeah, inside you before it comes out. Yeah, it's sterile. So ladies, the hoverers amongst you all, and ladies, you know what I mean. Sit down. Because those of you who hover, pee all over the seat. Disgusting. Yes? Okay, wipe the seat off. Sit down. It's sterile. When you take microbiology class, one of the things you need to do is go swab the toilet seat. That's not where you're going to grow bacteria. Guess where you're going to grow it. On the handle, walking outside the bathroom. Your urine is sterile. So relax, ladies. Sit down. But passing urine through your urethra is going to help flush out any substances that might climb up there. Another thing for women, you have a very short urethra compared to men. That's why you get urinary tract infections at a higher rate than men do. Are you the one that says, oh, I'll wait, because I'm busy. I don't go when I need to go. When my body tells me I need to urinate, I don't do that because I have too many things to do. Guess what? Pee when you're supposed to pee. Because again, that's going to flush the urethra and get rid of any foreign substances that might make it up that short tube. So if you're a woman that tends to get urinary tract infections, that might be why. Drink more water and pee when your body says, I have to pee. You'll help yourself. Second line of defense, cellular. So phagocytes, those natural killer cells, that inflammatory response we talked about, and certain chemicals that our cells give off. We're going to talk in a moment about one called interferon, and this is going to help protect us from viral infection of cells. And then another group of proteins, which we're going to talk about in a moment, called complement. And complement is a group of proteins that is normally just kind of hanging around in the circulatory system in fluid form. When it's activated by some of the responses that we're going to talk about in this chapter, it can also poke holes in things. So the first antimicrobial protein that we're going to talk about, which is this diagram, page 771, and that's figure 21.5, is an example of how interferon that's secreted by cells that have been infected by virus to help protect other cells. How do viruses work? Anybody know? Why are they such a pain in the neck? Well, they actually take over your DNA. So when a virus infects a cell, your cell is called the host, because viruses are like parasitic. So way up there at the top of the diagram, we see a nasty little virus. And a virus is usually depicted as some sort of protein outside, and then some little squiggly in the middle. You know what the squiggly in the middle is? It's genetic information. So that's basically all a virus is. Some sort of protein outside and some sort of genetic information inside. Now, for viruses, that genetic information can come in many forms. It can come in form of DNA. It can come in form of single-stranded or double-stranded DNA. It can also be in the form of RNA. So it depends on the virus. But a virus needs some sort of door to enter your cell. And usually it's one of the bazillion proteins that you have on the surface of your cells. It hooks on and then injects its genetic information into your cells. That genetic information is then going to go and take over your DNA's transcription and translation abilities. And guess what it's going to cause you to do? Make more of them. But when it does this, the cell sort of sends out a warning signal. It knows it's infected, it knows it's a goner, but it wants to tell a neighbor or help a neighbor survive. So it sends out chemicals called interferons. And if we look down here, they look like these little triangle bits. And interferons are going to help other cells. Remember, this cell's a goner. He's infected. There's nothing he can do. All he can do is just do what the virus tells him to do until the virus either kills him or goes into remission. But he can save his neighbor. So by secreting interferon, interferon is going to sort of block that door in. Kind of lock the door that that virus used to get into the original cell. It's going to interfere with the receptor so that virus can't go and attack the next door neighbor. So why do viruses make us sick? Well, because they've attacked enough cells that are taken over and cause them to make more virus to make us sick. But eventually what happens? It doesn't adapt to it. It's always going to be foreign. It can protect other cells from being infected, so it can slow down production. And then it's going to, and we're going to talk about specific immunity in a moment, your body is then going to make specific chemicals to fight off that foreign invader. What are those chemicals called? Antibodies. So this is one way to slow down the production of virus. By producing interferon, interferon is going to help ensure that other cells in my body are protected and help me to concentrate on the nasty little critter that got in, the virus. The other group of chemicals that are very important to help protect us is something called complement. Complement is normally a whole bunch of little proteins that are floating around free in liquid form in the blood plasma. And several different things can activate complement. Do you ever watch some of these superhero shows? You know, the bat light goes up in the sky and that activates who? Batman. some of the newer superheroes I'm unfamiliar with, but something changes and we press a button and transform into something else. That's kind of what happens with complement. Complement is going to be transformed into something else in several different ways. So when antibodies are on cells, target cells, that's going to activate complement. Something called the lectin pathway, when microorganisms and some of their surface substances are present, that can activate complement. We can also spontaneously activate complement. Again, microorganisms and their surface chemicals can do this. When an antibody is attached to its specific antigen, that can activate complement. And when complement is activated, it's sort of like pushing the superhero button. We take all of those separate little proteins, and we see them listed here, and we create a complex. Guess what the complex is called? What's MAC stand for? Membrane Attack Complex. A MAC attack. Yes, that's what it's called. I'm not kidding. Okay, so these proteins are going to band together and form a membrane attack complex. So activated complement components, and they put a C in front of different numbers basically to delineate all of the different proteins, can then insert into the membrane of foreign invaders. If I take a big, huge complex and poke it into you, what's going to happen to your innards? They're going to spill out and you're going to die. So complement fixation and the formation of a membrane attack complex, or a MAC attack, can also help to rid your body of foreign substances. And this is kind of what happens once that membrane attack complex has been formed. So we see all of the different pathways outlined for us on page 772. The classic pathway, the lectin pathway, and the alternative pathway. All of these are going to lead to the same thing, a membrane attack complex, which is going to be able to poke holes in things. So we have a bunch of people or a bunch of players in this immune response that can poke holes in things. Remember our natural killer cells, this membrane attack complex. And then they also talk a little bit about fever, increased temperature. Increasing temperature can denature foreign proteins. So are you the person who gets a fever of 99.9 and quickly runs to the medicine cabinet and pops Advil or Motrin? Yeah, don't do that. Why? Why do you get fever? It's doing a job. Now, when that fever goes way too high, that's when you should pop the Advil and the Motrin. But when you have that low-grade fever, stay home, chicken soup, Kool-Aid, that's the sure cure. Don't laugh, it's true. When you're sick next time, remember, chicken soup and Kool-Aid. That's it. You don't need medicine. You can have orange juice, anything with sugar in it, because you want to give your body more what? More energy for this attack that it has to. Yeah, Gatorade. If you lose a lot of fluid, something with electrolytes. Gatorade, careful with the Gatorade because it's got a little too much sugar, but like electrolyte supplements, Pedialyte, things like that, those things are good because they're going to allow you to fight off this foreign invader. So understand the different pathways for complement fixation, that MAC attack and MAC formation, and understand that chemicals given off by cells actually cause your temperature to rise, and those chemicals are called pyrogens. Those temperatures are going to help fight foreign invaders. So next time you have that low grade, let it go. I know it's a pain in the neck because you feel crappy, but it's there for a reason, and it'll help you get better quicker. Now comes the fun. Now we're going to talk specific. So we know all the players in a non-specific immune response, yes? Now we're going to talk adaptive defenses. So what is it exactly that we're defending against? How do we know it's foreign? It's got those proteins on the surface. I keep talking about proteins on the surface, proteins on the surface. Well, viruses have them, and bacteria have them, and every living organism that might make you sick will have them. You have them, right? Okay. So those proteins are antigens. And antigens, how did we define them earlier? Yeah, exactly. Antigens are proteins that can elicit an immune response. Basically be recognized as what? Foreign. Now you have self-antigens, but your cells already went to school, right? Your T cells know which ones belong to you because they went where? To learn. They went to the thymus. And what did they become there? It's a big word, immunocompetent. So those cells can recognize when someone comes in that is not self. And they can do it several different ways. They do it by something called antigenic determinants. Now, I use this as an example when I describe an immune response all the time. Does everybody have that sister-in-law they just love? Well, guess what my sister-in-law's name is? Jen. And guess what my children call her? Auntie Jen. Perfect! So how do I know when Auntie Jen is at my house? I get a fever and she makes me sick. No. How do I know it's her? And not just any old person knocking at the door. How do I identify Auntie Jen? There's something about her that tells me it's her, right? Her face. Yes? Some specific part. Now, I could chop her head off, and I wouldn't know it was her, would I? Because she's got arms and legs and kind of, there's a whole bunch of people that could be her. But it's that specific thing about her that makes me recognize Auntie Jen. And that's what I'm talking about when I talk about antigenic determinants. So any given antigen can have several, and most do, little portions of them that allow our cells to recognize them as foreign. In this particular diagram, we have 3 different, what? Yeah, antigenic determinants. Something on there that tells your cells that this is a foreign substance. So we see, in this particular case, this antigen that we're talking about has 3 very specific determining factors that our cells have the ability to recognize. No, antigen, blood type antigens also have antigenic determinants. Now, in order for me to neutralize and fight them off, those determinants are going to be important. Because I, in an adaptive defense, can make very specific chemicals called antibodies that will attach to these determinants. And just the determinants. I don't care about the rest of it. When antigen comes, I know she comes because I can see her face. And that's all I need to know it's antigen. Yes? It's the same thing with antigens that come into your body and invade. Your cells can recognize that determinant, antigen's face, and know it's a foreign substance. So, antigens have antigenic determinants. Some of them have several, most of them do. And your body can make very specific chemicals to neutralize them called antibodies based on their antigenic determinants. So for this particular antigen, how many different antibodies can my cells make to neutralize it? 3 different ones in this example. But those antibodies that are made are very specific, and they will kind of link in and lock in to cause an antigen-antibody complex, clumping, like we saw when we did our blood typing. The clumps are not going to allow those cells to just float around free because they're sort of clumped up in a complex. This is going to signal some of our non-specific guys to get rid of it. So when antibodies attach to antigens, our non-specific defenses, those cells that we talked about, can actually see something foreign doesn't belong. What am I going to do with it? I'm going to attack. So I'm either going to phagocytize it, or I'm going to run over with my little spear if I'm an NK cell and poke holes in it, or if I'm complement, I'm going to see that there's a big complex there, and I'm going to throw a little membrane attack complex on it and poke a bunch of holes in it. You see where I'm going? But antibodies are very specific. They will only attach to their very specific antigenic determinant that they were created for. So they talk a little bit about complement antigens and haptens. Not all antigens are complete. So like Auntie Jen, when she comes to the door, she's complete. She's all set. She's going to make me sick, right? But some things need a helper. Anybody allergic to poison ivy? Anybody not allergic to poison ivy? Okay. We're all humans, right? Why are some of you allergic to poison ivy and some of you not? Because the antigens are not complete antigens. They actually need a protein to hook into them to actually cause them to aggravate your system. These are called haptens. So they talk a little bit about incomplete or hapten proteins. And poison ivy is a good example. So if you don't possess the protein to make it a complete antigen, it's not going to cause you any problems. So all of you who are not allergic to poison ivy, you don't have the right protein. Actually, I would say you don't have the right protein, which is good. Not good for all the rest of us who are aggravated by poison ivy because we have that protein complex that we produce to cause it to be a complete antigen. In order for me to elicit an immune response, have people recognize this as foreign, have what we call immunogenicity, I have to be a complete antigen. A complete antigen is going to also have what we call reactivity. Again, be able to alert all the players in an immune response that I'm there so I can get rid of this foreign substance. So remember, in order for me to recognize antigen, I have to be able to recognize what? Her face in the case of my sister-in-law, but her antigenic determinant. So that's kind of what her face is.
So what do you do when she gets to determine like natural killer?
I go get my spear out of the closet? No, I have to be nice. So that's an antigenic determinant. Now remember, you have self-antigens, antigens on the surface of your cells that tell your players in an immune response who you are. That's not all they do. They're also going to help with an immune response as well. In this diagram on page 775, it talks a little bit about lymphocyte development, maturation, and activation. So how is it our cells understand what self is? How do they know what self is? And how can they protect us from foreign invaders? Well, remember, the lymphocytes are the players here in adaptive immunity, correct? And there's two groups of lymphocytes that are going to help very specifically prevent disease in my body. We have B lymphocytes and we have T lymphocytes. Now, some of those lymphocytes, which ones, go to the thymus? T lymphocytes. They're going to go to the thymus and learn about which antigens are self. Again, they're going to become immunocompetent. The B cells are going to end up there as well, but where do they originate from and kind of hang out and get their lessons? In the bone marrow. So the maturation process is a little bit different for the T cells versus the B cells. B cells are going to mature in the bone marrow. T cells are going to migrate to the thymus before they make their final resting place in the lymphatic system. So understand the difference between the two in the maturation process. Then we're going to find them hanging out in the lymphatic system, in those lymph nodes, in that spleen. And this is where they are going to be what we call the seeds. Okay? What's that? Spleen. Largest lymphatic organ, yes. So, antigens are going to encounter these cells through their travels. So, when I get a nasty virus in my body, when antigen comes to visit, she's going to take over cells and start reproducing, eventually making more and more of that foreign invader is going to get into my tissue fluids. This is where my cells are going to encounter those foreign substances. See her face. All I know as a cell that substance is foreign, non-specific. But when we talk about a specific response, I know her by face. I know that antigen by its antigenic determinant. When I see that, I'm going to go into action. When I go into action, again, the B cells and T cells are going to have a little bit of a different role with respect to fighting off a foreign substance. This is the last step here. So once I encounter the antigen, once I see it, and other cells in the immune response are going to help me see it, they are going to activate me, and I am going to then change. With respect to B cells, I'm going to change. I'm going to make more of myself, first of all, because in order for me to fight off a foreign substance, I can't be an army of one, right? I got to make a whole bunch more. So that's where proliferation and differentiation comes in, the final step. Once they've been activated, once they encounter the antigen, they are going to kick into gear and multiply and make more of themselves. They are going to do two things. Some of them are actually going to fight this war, go and specifically fight this war. If I'm a B cell, I'm going to make more of myself, and some of the cells are going to turn into plasma cells. That's the differentiation part. And those plasma cells are going to make what? Who? Antibodies. Very good. Those are the guys that are going to make that specific antibody. Specific to what? The antigenic determinant, correct? T cells are going to be the cruise directors of this whole response. They're going to help organize not only the B cells, to some of them turn into plasma cells and make their antibodies, but they're also going to help organize all those other guys we talked about in non-specific defense. Tell them where to go. Tell them who they need to get rid of. So there's cells involved in this process called antigen-presenting cells. So they're actually going to be like directors. Now, what was it that I recognized about antigen again? I knew it was her at the door. Just her head, right? So what if I chopped her head off when I encountered her, as an antigen presenting cell, grabbed it and put it out for everybody to see and say, as a T cell director of an immune response, this is what you have to go fight. When you see this, you go kill. Yes? That's what the T cell does. The CD4 positive T cell is going to help organize this whole response. And the CD8 positive T cell is actually going to go in and do the fighting. So under proliferation and differentiation, we have two separate players here, B cells and T cells. We're going to go fight off antigen. The reason they know who she is because, of course, the B cells came in contact with her, and then the T cells helped tell everybody else what they needed to fight off. Yes? So it's important that all the players in an immune response be intact. Self-antigens, MHCs, major histocompatibility antigens, are the reason all of these guys can do what they do. They're sort of like docking stations that help in an immune response. They help our cells recognize us as self. They're sort of like our signs on our cells that say, no, this cell is mine, don't kill it. And they are going to help teach the cells that run around and recognize foreign substances what to look for as self. So in this diagram, we talk about the actual selection process, and this is on page 776, figure 21.9. This is in the thymus. This is the education of the T cell. And we're going to use these MHCs to help educate the cell as to what self is. If for some reason I fail to recognize self, if for some reason I come in contact with my self antigens and I attack, do I want me around? No. I want me to be able to recognize self antigens. Failure to recognize self-MHC antigens is going to result in death. Because those cells that hook onto MHC tightly, we don't want them around. Sometimes the system goes wrong and they can create a group of diseases that can't recognize self-antigens. Those diseases are called autoimmune diseases. It's where these cells kind of flunk kindergarten. They don't make it through, but they made it through. So for some reason, they didn't get destroyed, or something changes in them to cause them not to be able to recognize self anymore.
I'm trying to think, my grandmother was in the hospital. She had, is it infection or, well, anyways, the antibodies and stuff they could give her just wouldn't work. Like your body resisted them.
I don't know.
She said we couldn't come in if were anyone sick or anything.
If you were compromised at all, because her body couldn't fight off. Okay, so for some reason her body couldn't produce the proper antibodies to fight off foreign substances. I'll give you an example. HIV virus. Yes? Does anybody know what that is? Now, HIV is a virus. HIV is a virus that specifically gets into cells using that little door we talked about. And the name of the door is CD4. The name of the protein on the surface of cells is CD4. Now, we find CD4 on the surface of which cells? T cells. Only certain T cells. The real important T cells that are going to help be the cruise directors to this whole immune response. When the virus gets into those cells, the immune response is running around blind because it doesn't know exactly what it needs to fight. It has a problem with specific immunity. That's why HIV is a problem. because it can result in problems with you being able to fight off the simplest of foreign invaders. And you might develop what from this? AIDS.
So can you have HIV in your system?
And not have AIDS?
Well, no.
Yes.
Without it attacking your T cells, though?
No.
Because the only reason why I asked, there was a case in Denmark, I want to say, the prostitutes are found that they're immune to HIV. How would that...
You can actually do what? The problem with viruses like HIV is they're inside your cells.
So can it be...
They might have some sort of substance that they're able to produce that will prevent them from getting inside the cells. Yeah.
Well, yeah, what I was thinking is because of those prostitutes, maybe they were exposed to it so much.
Just happened to be their genetics. They were lucky prostitutes. Okay? So you're immune to something when you can fight it off. Okay, so whatever, I'm not familiar with that particular case, but it might be that they have substances in their body that prevents the virus from attacking their cells. So, we have positive selection and negative selection. If we don't pass the test, if we can't recognize self, what's going to happen to us? Apoptosis, self-destruct, cell self-destruction. And hopefully in general biology you learn that term. When you talked about cell cycle, you learn that term. So cells will be initiated into apoptosis. If they pass the test, where are they going to go? If they pass the thymus test, if I become immunocompetent, I pass kindergarten, where do I go? I'm going to go to the lymphatic system and hang out there. So they talk about seeding secondary lymphoid organs, and they talk about becoming immunocompetent, and then encountering antigens. So remember, we encounter antigens many different ways. They can bump into us, all of those fluids floating around in the lymphatic system. We can encounter them in the circulatory system. Some of the cells have the ability to spread out arms, like this one here called the dendritic cell. And dendritic cells are going to help protect us from foreign invaders as well. We're going to find them throughout the body, and they have the ability to initiate an immune response. So on page 777, we see them described as the body's frontiers. And we're going to find them in things like skin. When we talked about skin, we talked about dendritic cells way down in the basal layer of the epidermis. Remember those cells? Go back and look in chapter 5. Who's got the book? Go look at that diagram of the skin. I think it's on page. Now I got to go look. Can't remember what page it's on. It's on page 153. Very good. See the purple cell? What's it called? A dendritic cell. And it's right there in the skin, watching out for foreign substances. We also have cells like macrophages. Macrophages, where am I going to find those? No, down in connective tissue. So we're going to find them in the liver. We're going to find them in connective tissue. And then we have B lymphocytes. Those B lymphocytes are going to help find foreign substances and then protect us by creating antibodies against foreign substances. So we see these guys described on page 777 under antigen-presenting cells. And these guys, combination of these guys, actually have the ability to engulf the natural predator, something foreign, and then do what I said, cut off her head and place it on the outside. Show everybody what they need to go fight. Present the antigen. And MHC proteins are that platform, so I can present the antigen to the rest of the immune players. So antigen-presenting cells, dendritic cells, macrophages, we find antibodies on the surface of B lymphocytes that are going to help activate them as well. And between all of these guys, we're going to help initiate an immune response. Okay, so adaptive defenses, humoral immunity. Humoral refers to what? Who? Fluids floating around in the body. This is where we're going to find these substances, in the humors, in the plasma, in the tissue fluids. How do we actually produce them? Well, first of all, we have to know what we're fighting against. We have to know that antigen is at the door. In a primary response, we're going to have that initial encounter. And that is going to cause cells to change, to differentiate and proliferate. In the case of B cells, we are going to activate them by bumping into antibodies for very specific antigens on the surface of these cells. Right now, as you sit here in this room, you have the ability to recognize as foreign any antigen that you will ever come in contact with in your entire lifetime, whether you come in contact with it or not. Every single foreign substance, because these cells have the ability to kind of shuffle their genetic material. I'm not going to get crazy about it. This is a whole study in itself. This is a whole science in itself in this one chapter. But you have the ability to recognize any foreign substance you will ever come in contact with. And it's because of these B cells that hang out and have become mature that sit in your lymphatic system with these very specific antibodies on the surface. Now, this isn't an actual number, but hopefully it helps you see how things work. Say you have one cell in every single lymph node that can recognize a foreign antigen. This foreign antigen has never come into your body before. Say it's antigen. I'm meeting her for the first time. That would be like the initial response. Once those cells become activated and see the foreign substance they've been waiting for their whole lives, what are they going to do? Well, they're going to start making more of themselves so they can chop her head off and fight her off. They're going to start to proliferate and differentiate. Some of them are going to become plasma cells. So we're talking B cells now. We're not talking T cells. This diagram is just the B cells. We're going to make more of our cells. Some of them are going to become plasma cells and make those very specific antibodies, the ones that can neutralize antigen. And then I'm going to make more. So remember how I only had one in every lymph node before? At the end of this, I'm going to have 20 in every lymph node. The next time antigen comes to invade, now instead of just one, I have what? 20. I have a big, huge army that can fight off this foreign substance. I'm always going to have antibodies hanging out in my humors for this foreign substance. Once I encounter it, though, I'm going to have enough of it to actually neutralize it. And when I encounter it again, what? I'm going to have a lot more fighters in the army that can produce much more antibody at a very fast rate. And that's what they talk about under secondary response. So years later, when antigen comes to the house again, or when something like chickenpox comes to your house again, the first time you encounter it, what does it do? It makes you sick. It makes you sick because you only have very few players in that initial encounter. But at the end of that encounter, you're going to have a lot more players in the war. So when chickenpox comes back to visit again, I'm going to squash it before it can even make me sick. You can only get it once. But it doesn't mean you don't encounter it more than once. You can only get sick by these guys once, normally. Sometimes your immune response doesn't do what it's supposed to do. Everybody's different. But remember, when you encounter a virus, for example, and this is where I'm talking about virus. Virus is very specific. You will never get sick again under normal circumstances from that virus, because you will have encountered it, created all of your memory cells. Those are the guys that are always going to remember them. Now I have a lot more of them, and you're always going to have enough antibody circulating. Now sometimes your cells kind of, if you don't encounter it for a while, they kind of get lazy. Sometimes we need to give them another little boost, make more of them. Like some of your vaccines, for example, you have to get what? Boosters. What are you doing when you give somebody a vaccine? You're creating this response, but you're doing it artificially. So it's like taking Auntie Jen, chopping off her head, sticking it on a pole, showing everybody. Everybody goes crazy, immune response, but the rest of her was never there to make me sick. So when you get your flu shot, what is it? It's causing all of this to happen. It's like holding the head on a pole. We take the antigenic determinant, we show it to your cells do all of this, but the whole intact organism isn't there to make you sick. They don't get sick from that. They get sick because I'm busy fighting a war. Somebody else comes in the back door. Yes? You don't get sick from the vaccine, but you do compromise your immune system, because your immune system, because of the vaccine, will fight the war, right? Somebody can sneak in the back end. Because your body's fighting off whatever is making you sick. We don't want to turn around and introduce something else to distract your body from fighting off what's making you sick. Make sense? So get your flu shots. Read a book called, I can't remember the title of it. It's a story about the 1918 flu, the Spanish flu. Scary as hell, and you'll never miss a flu vaccine again. Okay? So, that's what your body is doing when you get a vaccine. So, activating B cells is discussed on page 779 in the humoral immune response. Remember, we're making chemicals, very specific chemicals. That's the B cell's job. So, B cells and T cells have two different jobs, and this is outlined for us in this nice little table on page 779. B cells is more humoral, creating chemicals, putting it in our fluids. They're going to make antibodies. What is their primary target? Whatever the pathogen is, the specific pathogen, that's their primary target. Where do they come from? Bone marrow. Where do they mature? Bone marrow. Which cells are going to cause the effect in the response? Those plasma cells, because those plasma cells are going to do what? Make those specific antibodies. They're going to fight off those substances. And don't forget, I'm going to make a heck of a lot more players the first time I encounter antigen. So now I have a whole bunch more. I have what we call memory cells. So an immune response has a memory, and the memory creates a large army. T cells, on the other hand, is going to be the cellular part of the fight. T cells are going to help get rid of debris that are neutralized by antibodies. So do T cells make antibodies? No, but they're extremely important because they are going to direct everybody as to who to fight off. So we talk about their primary targets being viruses and all of those other things. Foreign proteins on the surface of cells elicit immune response. And when you start making foreign proteins on a cell like a cancer cell, your cells are going to go in and destroy it. Remember one thing, you can always fight the war. Do you always win? No. So you can take that into the cancer realm as well. My cells are going to recognize those cells as foreign, but sometimes it doesn't happen until we've made way too many of them, and they're harder to fight off. When you get that flu vaccine, if something else makes you sick, it actually takes advantage of the fact that you're fighting a war. Right? So remember, we have originated in the bone marrow, but I'm educated in the thymus, and these are some of the guys in that category of T lymphocyte that are going to help get rid of those foreign substances. So cytotoxic T cells, helper T cells, regulatory T cells, all of these guys have the ability to kind of poke holes in things and get rid of the trash. The same thing's going to happen to the T cell family when we encounter a specific antigen. We're going to make a lot more memory cells. So the next time this foreign substance comes along, we are going to be so ready for it, it's not even going to make us sick. On page 779, we see this diagram. This is a nice diagram because it's a visual example of how much chemical we actually have in the body when we encounter foreign antigens for the first and second time. So say it's chickenpox, because everybody kind of knows a little bit about chickenpox, although you younger people aren't having the thrill of experiencing chickenpox like us older folk did. In my day, somebody had the chickenpox, everybody brought their kids over because we wanted them to get sick and be done with it. Yes? Now you guys get vaccinated, oh, you're all such wimps. So, chickenpox. First time it comes. It's kind of like the first, let me see, Virus Sharon. Let's call her Virus Sharon. That's my mother-in-law. Since we're on the in-laws, what the heck, right? So Virus Sharon comes to the house. First encounter. What's going to happen? I'm going to recognize it as foreign. I'm going to rip some of it apart. Some of my antigen-presenting cells will go, go fight this! Yes? My B cells are going to kick in. My T cells are going to kick in. Everybody's going to get organized. Everybody's going to fight. So between 7 and 10 days, and I bring this up because that's the kind of the chickenpox thing. When do you get most of your little bumps from chickenpox? Between 7 and 10 days. Not right after you're first exposed. It takes a while for those viruses to go pick on all of your cells, right? So around 7 to 10 days, we start seeing the effects of something making us sick. But remember, our body is fighting that war as well. So around 10 days is when we start making enough antibodies from this response, so we start neutralizing the antigen. So even though it's all over the place and making me sick, by about 10 days, my cells in the immune response have made enough antibodies to start neutralizing the antigen. And what happens after that time? Chickenpox. You get the bumps, you scratch, you go take the bath, but then what happens? They go away, right? How come? Because now your body, those fighters in the war, have made enough chemical antibodies, very specific to Virus Sharon, to neutralize. Yes? What else did I make besides the antibodies? I made all my fighters in the war. I made a whole bunch of memory cells, right? And those memory cells are hanging out, constantly surveying. So the next time Virus Sharon comes to visit, on day 28, what's going to happen? The same exact thing, except now I have tons more players. Now I have tons more cells because I created those memory cells. So now when I create my antibodies, hell, it's not going to take me 10 days. It's going to take me 2. And I'm not going to make just that much antibody. That much antibody can actually neutralize the invader. I'm going to make that much antibody. Are you going to make me sick? No. I'm going to squash you right from the beginning. Because that secondary response, after I've been exposed to that virus, Sharon, is so much more violent. I'm going to make so much more antibody that I'm going to neutralize those guys before they can get a chance to build up enough to make me sick. That's why you don't get sick again. I've been sick all winter. Guess what? Not from the same thing. Unfortunately, because viruses are so simple, they also have the ability to change. So it's kind of like virus Sharon or antigen having the ability to undergo plastic surgery very quickly. So even though it's still them, I can mutate and change enough so the antigenic determinant is kind of like something I've never seen before. Yes? So there's so many different viruses out there. They have the ability to mutate at a very fast rate and change enough so that your body can't recognize them. And then what does your body have to do? Same thing all over again. When you're little, your body doesn't have a lot of these guys all built up. Yes? So you tend to get sick a lot. So eat dirt, sit on the toilet seat, don't use antibacterial soap every single time you turn a corner. Yeah. Build up your what? Build up your immune system. And your exposure to simple things on surfaces has you undergo an immune response. You're old now, so you've been exposed to a lot of these things. Remember when we talked a little bit about tonsils earlier in this chapter? I said at a certain age, it's going to seem like the tonsils are always swollen. How come? We're constantly doing this to brand new antigens for that person's system. But once you've developed all of these guys, they're all hanging out in your lymphatic system. So a healthy operational immune system has so many different fighters that have been exposed to these foreign substances and have players that can fight them off. So, how do I develop immunity? And they also talk, don't forget, we have an immunological memory. Those memory cells are going to be able to produce lots more antibodies for us. So, there's 2 different ways we can fight this war or initiate this war, let's say. So we have humoral immunity. And what was that? I have antibodies floating around in my body fluids, in my plasma, in my tissue fluids. Remember how I got it? I got exposed to some foreign substance. I made my cells, they differentiated, they proliferated, and they did all of those things we mentioned earlier in the class. Now there's 2 ways to do that. The old way of getting the chicken pox, which is what? Active. That means I come encounter the chicken pox virus. My mother brings me to my friend's house. I get all of my other little friends sick with the chicken pox virus. Yes? So that's naturally acquired. I get sick. I get the infection. I get chicken pox. I undergo the war. But we can also actively do that with the antigen artificially. And that's what your vaccine is. So it's like me, again, chopping Auntie Jen's head off, sticking it on a stick, and showing everybody this is what you need to fight. Giving you the antigenic determinant in an inactive way. So the organism isn't going to make you sick. So it's either dead or attenuated, kind of the same thing, kind of half dead, not having the ability to take over your cells and make you sick. but doing the exact same thing. So when you go for your flu vaccine, what is it? Yeah, it's attenuated or dead actual proteins, antigenic determinants on the surface of whatever that are going to cause an immune response to occur. I want to do this quick before, oh, wait a minute. All right, so let's take a quick clicker question. To intrinsic, what's intrinsic? Inside the body, right? Defense systems have many components. Which of the following cell types is not a contributor to specific immunity? Specific. And that's A, B, C, and D. Five, 4, 3, 2. There's only 27 people in class today? 28? 29 maybe? No? We done? What's the answer? It's a natural killer cell. All the other cells we just talked about, plasma cells, B cells, T cells, all specific. All right, let's go back to my discussion. So, that's active immunity. I either get sick or I get a vaccine. I get the actual antigenic determinant. I can also protect you without you getting sick. Unfortunately, doing it this way, you're not going to elicit that big war. Okay? You're not going to make all those cells. But I can protect you. So if I give you the antibodies, and we can actually give you antibodies in mother's milk, or through the placenta, or I can give you something called the gamma globulin shot, a whole bunch of antibodies to protect you from a foreign substance. It's going to do the exact same thing your natural antibodies would do, neutralize the antigen. Your cells are going to go clean up the mess, but you're never going to do what? You're never going to fight the war yourself. So you're never going to make memory cells, and you're not going to have a whole bunch of antibodies floating around in your circulatory system. So we can either do it natural through mother's milk and all of that good stuff, or I can actually give you an injection. So we can do it artificially. But in passive immunity, you're not going to create memory cells, and you're not going to have a high titer of antibody. When they go to see if something worked, like you probably all had to do this to come to school, right? You had to go get a titer to make sure your MMR worked. What's an MMR? Mumps, measles, and rubella, all nasty little critters that you got vaccinated for. And they said, no, go to school if you don't have enough antibodies in your system to fight these buggers off, because we don't want you guys to make everybody in school sick. Yes? So what do they actually go in and do? No showing the heads or anything. They're actually going to go test your humors to see if you have enough of what in there? Enough of the antibodies to fight off these antigens. That's what a titer is. So I'm going to go in the lab and I'm going to see whether you have enough antibodies to neutralize the antigen in question. If you don't, what am I going to do? I'm going to go give you another shot and make you make enough antibodies. I want you to be safe from that disease so you don't get it and make everybody else sick. Yes? So know the difference between active and passive immunity. understand what humoral immunity is. We're going to talk about antibodies next class, but antibodies are the actual chemical substances. So we'll pick up with that on Thursday. If we're not here Thursday, what are we going to do? We're going to go listen to Maria finish the lecture online. And I won't put an old lecture up there. I'll make a whole brand new one. Okay? And what does that mean? You're going to also look for another quiz. Yeah, and don't forget to look for your essay hints.